Assemble the equipment. Sanitize your hands and put on gloves. Place the capillary tubes in the centrifuge opposite each other, with the sealed end outward against the outer rim. Note the numbers of the locations. Record the location of the tubes in the centrifuge log sheet. That way, if you're spinning samples for more than one patient, you'll be able to tell them apart. Place the locking top over the threaded bolt on the centrifuge head and turn the fastener until it is tight. This will prevent tubes from moving out of the grooves while they're being spun. Close and latch the lid. Spin for 3 to 5 minutes at 10,000 RPM. Most microhematocrit centrifuges are preset to the correct speed. If so, set the timer for 3 to 5 minutes. Wait until the centrifuge comes to a complete stop, then open the lid and remove the locking top. If the centrifuge contains a reading device, and your capillary tubes were filled exactly to the calibration line, adjust each tube so that the bottom of the red blood cell column is at 0. This is the point where the column meets the sealing compound. Note the result on the reading device at the top of the red blood cell column. If the results from the two tubes do not vary by more than 2%, average them to get the final reading. If your centrifuge does not have a reading device, or if you used capillary tubes without a calibration line, or if you filled the tubes above or below the calibration line, you will need to use a hematocrit reader. Place the first tube in the hematocrit reader with the junction of the sealing compound and the red blood cell column at the 0 mark. Rotate the outer disc to the 100% line. Then rotate the inner disc until the reading line is even with the meniscus of the plasma. This defines 100% of the entire sample. Now rotate the outer and inner discs together until the reading line is at the junction of the red blood cell layer and the white blood cell layer. Record the number on the outer disc. This is the hematocrit result, the percentage of red blood cells in the sample. Repeat this process with the second capillary tube. Record the number on the outer disc. If the two numbers do not vary by more than 2%, average them to get the final reading. If they vary by more than 2%, you will need to repeat the test. Discard the capillary tubes in a rigid biohazard sharps container. Remove and discard your gloves and sanitize your hands.